Hello, my name is Prashant Hariharan. I'm a PhD candidate in the Department of Biomedical Engineering at Wayne State University and a recipient of the Graduate Fellowship from the International Foundation for Ethical Research. The aim of my project is to develop an organ-on-a-chip model of the choroid plexus, the part of the brain that secretes cerebrospinal fluid. My objective is to advance the understanding of hydrocephalus, a neurological condition characterized by the buildup of cerebrospinal fluid in the ventricles of the brain, causing an increase in pressure inside the skull. Current treatment strategies focus on surgically diverting fluid from the brain, and while these interventions improve symptoms, they do not address the underlying causes of hydrocephalus. No successful non-surgical interventions exist because we do not yet understand the mechanism by which the choroid plexus secretes cerebrospinal fluid. The animal models presently used are expensive, plagued by variability, and difficult to translate to humans due to species-specific fluid secretion mechanisms. In vitro systems, on the other hand, do not capture the complexity of functional human tissue. There is a critical need for a human-relevant in vitro platform to study cerebrospinal fluid secretion in the context of hydrocephalus. My project aims to address this need by engineering a novel choroid plexus on a chip. I use 3D printing to fabricate molds and rapidly prototype iterations of the chip design. Once the chip is assembled, the blood-cerebrospinal fluid interface can be recapitulated by growing choroid plexus epithelial cells and endothelial cells on opposite sides of a membrane. I use primary human cells to make my model translatable. The organ-on-a-chip system can be connected to syringe pumps to provide growing cells with the mechanical cues of fluid shear and pressure. Electrodes incorporated into the chip track the integrity of the barrier as a function of the ions transported across the membrane. Once the cells have formed a barrier, I can expose them to inflammatory triggers and test my hypothesis pertaining to the loss of choroid plexus barrier integrity and deregulation of fluid secretion. The generous funding from the IFER Fellowship will help develop, test, and validate a human-relevant model of the choroid plexus that has the potential to replace currently used preclinical animal studies. This could open the door to identification of novel targets, screening of new drugs, and the development of pharmaceutical treatment strategies for hydrocephalus. I'm very grateful for the support from the International Foundation for Ethical Research, and I'm excited to make my contribution.